Today we're going to be working on looking at just how specific and how susceptible enzymes are to changes in substrate as well as changes in pH or temperature. Now remember, the most important thing about organic molecules, and it doesn't matter which type of organic molecule you're talking about, whether you're talking about carbohydrates, lipids, proteins, or nucleic acids, remember one of the most important aspects of that organic molecule is its shape. Shape is all important when it comes to how these organic molecules function. None more so than proteins, especially enzymes. Enzymes are extremely specific based on their shape. So what we're going to look at today is just how specific are they. We're going to use two very similar sugars. Now, these particular sugars are disaccharides. Now, as you remember, di meaning two, saccharide meaning sugar. Each one of these disaccharides, one being sucrose, a plant-based sugar, and lactose, which is more of an animal-based sugar you find in milk. Each one of them contains the monosaccharide glucose. So how are they different? Well, if you look down at your lab, you'll see a kind of structural diagram of both sucrose and lactose. You'll notice that sucrose, the disaccharide, is glucose and a fructose monosaccharide, joined together, of course, by dehydration synthesis reaction, which puts them together. And lactose is a disaccharide comprised of glucose and galactose, again joined by a dehydration synthesis reaction, which put them together. Now, as you know, in order to split that bond that's joining those disaccharides, we're going to need to add water, which of course would be a hydrolysis reaction. But as you know, all chemical reactions require energy. Now, if you add an enzyme, it greatly decreases that activation energy that we talked about in class. So what we're going to be using today is an enzyme designed to break down lactose. Well, what's it going to break it down into? Well, hopefully it's going to hydrolyze that bond, and we are going to end up with glucose and galactose. But we're also going to see, can it break down sucrose? Because sucrose is pretty close to lactose. They both have glucose. Maybe it's close enough of a shape that it can react with it. So what would we be looking for? If this enzyme was capable of hydrolyzing that bond by catalyzing this reaction so that water is able to hydrolyze the bond between, say, in lactose, the glucose and galactose, or if it was able to hydrolyze the bond between glucose and fructose in sucrose, both of them we would be able to find glucose. So if we had a way to test for glucose, we could use that to determine whether or not this enzyme was working. Just so happens we do. There are glucose test strips. Now typically you find these in like a pharmacy. Because one of the things that often occurs with people who have certain diseases or conditions such as diabetes is that the glucose can be released into urine. So these are urinalysis test strips. And what they do is they look to see, is there glucose present in a solution? And we're going to use those today. So what we're going to do is we're going to take a look at a couple of different solutions. The first solution we'll look at is milk. And the second solution that we will look at is sucrose, or just sugar water. And to that we are going to add an enzyme, lactase, which should, remember, based on its name, lactase should catalyze the reaction of lactose. But we're also going to see, can it catalyze the reaction of sucrose? Then we're also going to take this enzyme and we are going to expose it to a large amount of heat. And then hope to see whether or not it will actually denature it. Now, if it denatures it, it shouldn't work, which means we shouldn't find any glucose. So we'll test that too. And just to make sure that nothing crazy is going, we are going to use a control. We're going to add a little bit of water to one of the milk. Even though milk is mostly water, we are going to add it just as a control. Because if glucose shows up in that, well, there's probably something wrong with our glucose test strips. So based on that, let's get started. So here we are again, and here's our setup. Now remember, make sure you're following along in your lab. The lab was designed for you to be doing this, but you're going to have to see it through my eyes. We have 5 test tubes. We have test tube 1, 2, 3, 4, and 5. In test tube 1, we're going to be adding milk and some water. That's going to be our test. In test tube 2, we're going to be adding milk and lactase. In test tube 3, we're going to be adding milk and our denatured enzyme. In test tube 4, we'll add our sucrose and water, again as a control. And in test tube 5, we will be adding our sucrose and enzyme solution. So for today, if you're curious, what we're going to be using today is this store-bought dairy relief, not plugging CVS, but there it is. And if you look here, you'll notice, and I'm sorry about that little shine on there, it says Lactase Enzyme Supplement. And you probably can't read it because it's kind of small print, but it says ingredients, lactase enzyme. So this contains a version of the lactase enzyme that normally your body would produce. So what I've done is I've taken that lactase enzyme and I basically crushed it up into a fine powder. So all we did was take one of these tablets, tossed it in our mortar and pestle, gave it a bit of a grind, reduced it into a powder, and added it into our beaker here with some water and mixed it up. Now, it's kind of chalky looking because when they make this, they add a binder, and the binder just happens to be fiber, good old cellulose. Next, we have some denatured enzyme. Now, it looks a little bit the same, it's just a little bit cloudy. What I did was take this, and I boiled the living daylights out of it. So in other words, I exposed it to a large amount of heat over a long period of time. And then you can see some of the kind of fuzz up here. When you boil it, I kind of scraped off the fuzz so you could see things a little bit easier. But that's all this is enzyme that has been heated up to a relatively high degree. And finally, we just have our water, which is just, just good old-fashioned water that you get from the sink. And that's going to be our control. So what we have today to test is this is our sugar water. It's just regular old sucrose, table sugar that you would get from a store. And for milk today, we're going to be using this amazing Glenview Farms, organic, nothing but the best for you guys. And as far as our test strips, these are the glucose test strips. So what happens is if you don't see any glucose, it should be of this yellowish color. And as the amount of glucose increases, if it can find it, it will turn progressively greener and greener. Now, if you're wondering what they look like, this is what the glucose test strips look like. And you'll notice that the tab here is yellow because it has no glucose at the moment. So the first thing we're going to do is take our test tubes and fill them up with, as it says, about 2 milliliters of either milk or sugar water. So what I'm going to do to add to each one of these test tubes 1, 2, and 3 are going to get milk. So I'm going to add approximately 2 milliliters, and yes, I am eyeing this. The exact amounts of milk will not matter too much as long as I'm fairly close to 2 milliliters. So I don't have to be extremely cautious with this. So I will fill each of these with approximately 2, actually it's going to be a little bit more than 2 milliliters. I'd say that's probably closer to 5 or so. But you can see that's roughly how much is filled in each one of them. And as I mentioned, these Why, no, I did not just spill the milk everywhere. If you were curious, I was just moving something. But regardless, we are back. So, in test tubes number 4 and 5, I'm going to add a roughly equal amount of this sucrose water, which again is just sucrose dissolved in water. So I will pour roughly the same amount in these 2 test tubes. And the next thing I'm going to do is do our additives. Now, again, I will point out that you should be following along in your lab, and if you're looking at the table on the 3rd page, we're about to start adding what it says. Now, in test tube number 1, it now has milk. So I'm going to add to that this water, about 1 squirt. Now, these are approximately 1 milliliter pipettes, but I'm going a little bit higher, so they'll probably end up getting about 2 milliliters of fluid in there. So I'm going to go ahead and add 1 drop or 1 squirt of water in test tube 1. Next up, we have the enzyme, which is just the lactase we looked at earlier. Again, this is dissolved in water. Again, I'm going to take up 1 squirt and put that in test tube number 2. Next up, this is the denatured enzyme. This is the stuff that I boiled pretty much into oblivion. And we're going to take a test tube full of that and stick it in test tube number 3. Here's test tube number 4. This is the sucrose. And again, we're going to use water as our control. We're going to add 1 squirt of water in there. And because we wanted to see if this lactase was really super specific, we're going to add 1 good squirt of lactase into this sugar water. All right, so let's review. Let's take a look at our test tubes. And I will try not to knock over the milk again. Not that that happened first, but yeah, I'm just saying. So anyhow, so here's test tube 1. Test tube 1, remember, has milk in it. So this has lactose sugar. And we added some water, just as our test. Here's test tube 2. Test tube 2 also has lactose, and it now has the lactase enzyme. Test tube 3, it has milk in it, so it has lactose, and we added the denatured, the shape-changed enzyme. In test tube 4, we have sugar water, or sucrose and water. And we added to that more water as a test. Here's test tube 5. Again, we have sugar water, sucrose, and water, and we added to that the lactase enzyme. So that should help you with your table. Now, we need to wait a little while just so these have a chance to kind of mix together and do anything that they might do. And we are going to wait a couple of moments.
A few minutes later.
And it's been approximately 10 minutes. I know, it seemed like it just flew right by. And what we're going to do is we are going to get out our test strips. Now, again, you'll notice that these strips are yellow at the ends because they have not been exposed to anything. So they are currently reading 0 glucose. And what we're going to do now is I'm going to take each one of these test strips, and I'm going to take the test tube and just tip it a little bit. And I've already poured milk, not that that happened, all over my table once. So I'm just going to carefully turn it to the side. And if you can see what I'm doing, I'm just turning it to the side and submerging it in the milk. And what I'm going to do is I'm going to put this right next to the test tube so I know which one goes with which. So again, if you're having trouble seeing what I just did, I apologize. I'll do another one for you. So I'm taking the test tube, I'm turning it a little bit so that the milk is actually kind of coming down, and I'm putting the test strip in the milk. And I'm just going to keep doing that until all the samples have been taken. All right, so what we have here are our test tubes with their corresponding test strips, and we're just going to give them a little bit of time just so that the material that's on each one of these strips has an opportunity to get exposed to the chemical that's in there. But I will move us in just a little bit closer so we can start to see if there's any sort of change going on. Now remember, what should be happening here is if in any of these test tubes we were able to hydrolyze, again, the name for the reaction, which is going to clip the two disaccharides apart. So we'll end up with either glucose and fructose or glucose or galactose, depending on what we're splitting. But we should see the presence of glucose if that hydrolysis reaction was successful. Now, we should be able to think a little bit, well, which one would we expect to have a color change? Well, in theory, lactase is designed to break down lactose. So I should be able to see it in there. And I'm not sure if you can start seeing it, but it looks like we may have some presence of glucose in one of these. So we'll take a look at number 1, number 2, number 3, and number 4. And if you want, I can probably get us in just a little bit closer and perhaps get us in a little bit more focus. So let's get started. So this is test tube number 1. This is test tube number 1, and this is the strip. This is test tube number 2, and this is the strip. This is test tube number 3, and this is its strip. This is test tube number 4, and this is its strip. And finally, this is test tube number 5, and this is its strip. So again, we'll bring these guys back, and if you look at them, you can figure out whether any of them changed color. So, now that we have these all reacted, this is the type of result that either we were expecting or not expecting. All right, so now that we've gone through the video, and it might have been a good idea to have had your lab open with you, I hope you did, and as we went through those particular scenarios in each one of the results, hopefully you have been filling in the table. Now using that information in the table, now you can go ahead and answer all the analysis and conclusion questions. And again, remember to use your knowledge of what you would expect an enzyme to do and how it's supposed to work to explain the results you got. Okay?